So let's get right into our webinar. We are so privileged today to have with us Bruce Blumberg, and from the University of California at Irvine. He is a professor in the departments of Developmental and Cell Biology, Pharmaceutical Sciences, and Biomedical Engineering. His current research focuses on the role of nuclear hormone receptors in development, physiology, and disease. He has particular interest in the early patterning of vertebrate nervous system and the links between exposure to environmental endocrine disrupting chemicals, development of cancer, and obesity. He proposed the obesogen hypothesis, which holds that exposure to chemical obesogens can reprogram metabolism to favor the storage of despite normal diet and exercise. And he has recently shown that prenatal exposure to such obesogens can have permanent effects on the exposed individual, their children, and their grandchildren. Dr. Blumberg was appointed as a Fellow of the American Association for the Advanced Science in 2002. I would just like to say that his advances in this area have been really important, have really opened up a whole new way of looking at the problem of obesity in this country, really advanced our understanding of disrupting chemicals. So it's with great pleasure that I turn the webinar over to Dr. Blumberg.
Thank you very much, Lynn. It's a great pleasure to be here and to be able to talk about our work to a much broader audience than I normally get to talk to. So what I plan to talk about in the next 30 minutes is a little bit of the evidence that there are such things as obesogens and the evidence that they may contribute to obesity. I'm going to talk a bit about our work that shows that obesogen action may involve the reprogramming of the fate of stem cells in the body such that they tend to become a different kind of cell than they would otherwise. And last, I want to talk about our work that shows that the effects of obesogen exposure may be permanent and passed on to offspring. So we hear that there's a worldwide obesity epidemic, and as I travel the world, I can tell you that it's primarily a U.S. epidemic, although people in other countries are getting heavier all the time, but it's really pronounced in the United States. So according to statistics, this number is now more than 35% of the population is what's called clinically obese. That means a body mass index greater than 30. That's twice the worldwide average, and it's 10 times the average in countries like Japan and Korea, which are also very developed countries. So it's not strictly a disease of developed countries. There's something more going on. The vast majority of us are overweight, and that's estimated to be 86% by 2020. So obesity has many consequences, and one of them is that it can cause international incidents. So here's a picture of Michelangelo's David, and here he is after he had a tour through the United States, which the Italians are very unhappy with us. Seriously, obesity is problematic because it adds a great burden to the healthcare system. And the most recent numbers suggest that approximately $210 billion in healthcare costs are added into the system due to diseases related to obesity, and that's fully 20% of what we already spend on healthcare. And as you can imagine, if that cost weren't in the system, that money could be diverted to other things, like, for example, providing healthcare to people who don't have it. The primary costs due to obesity come from its association with metabolic syndrome and type 2 diabetes, cardiovascular disease, heart attacks, strokes, also cancer, lost days from work. There's a variety of ways that obesity adds costs to the healthcare system. This is a slide from the Centers for Disease Control showing just what's happened in the United States in basically one generation. So in 1990, less than 20% of the population was obese. It was more or less 15%. And now, as recently as 2008, the majority of states are showing high rates of obesity. And actually, the Colorado is showing blue there, but it's also gone over to the red side in the most recent statistics. I use this number, this pictures, because they go together with another number that's also approximately doubled over the same time period. And this is contrary to what we constantly hear in the media. This number in the red boxes represents the number of health clubs in the country adjusted for population. So either these health clubs are empty and people are just building them and no one goes, or people are trying very hard to not be obese. And that's the opposite of what we hear. And what we hear is that we're obese because we eat too much and we sit around in front of the computer and the television and we don't exercise, and that's the whole story. Positive energy balance. Calories in, calories out. All calories are the same. We eat too many calories, and that's why we're obese. Here's a classic example of someone who eats more calories than he burns, as a gigantic sumo wrestler. But the fact of the matter is that there are other factors in obesity besides simply calories. The first is that we know that stress, chronic stress, which elevates glucocorticoids, causes increased risk of obesity. We know that not getting enough sleep, which may or may not be related to stress, is also linked with obesity. People who do shift work, who work at nights, who work the late shift, have a whole host of health issues, among them obesity. There's a model that there are such things as thrifty genes, which evolved years ago when we were hunter-gatherers to help us to make the most out of calories, which were scarce. There were no supermarkets. If you wanted to eat, you had to go out and collect your food or dig it up or kill it. So genes which would enable you to store more of the calories that you consumed would be evolutionarily favored. We know that there are such things as viruses like adeno-associated virus 36, which increases the risk of obesity. Very significantly, we know that the bacteria in your intestines are related to obesity. So the ratio of a kind of bacteria called Bacteroidetes to another kind called Firmicutes in your intestines predicts whether you'll be obese or not. And if you take bacteria from obese mice and put them into lean germ-free mice, those lean mice become fat, and vice versa. We know that there are changes in, we call them single nucleotide polymorphism, single changes in the DNA sequence in 30 or 40 genes that are all associated with obesity. We also know from extensive studies from the University of Southampton that prenatal nutrition and the in utero experience is associated with later life consequences. If mom has too much nutrition or too little nutrition when she's pregnant, the baby will have a propensity towards obesity later in life. And perhaps most telling, we know that maternal smoking during pregnancy causes decreased birth weight and increased risk of obesity in 100% of studies that have ever been done around the world. And there was a paper about 10 years ago that suggested perhaps there is a role for chemical toxins in obesity. And then the author showed, roughly speaking, that the obesity epidemic correlates with the increase in the use of chemicals since World War II. And sure enough, it does. But as I showed you, it also correlates with the number of health clubs. And you can make lots and lots of correlations between obesity and some event that's happened over the last 50 years. It's our job as scientists to try to understand not what are the correlations, but which of those might be causal. And the reality is that a subset of the chemicals that have come into use in the past 50 years have effects on the endocrine system. So as you may or may not know, weight in the body is under the control of a variety of hormones. And perhaps the most important ones are leptin, which controls energy balance, and ghrelin, which controls hunger. The thyroid hormone receptor, which is a member of the family of receptors that I work on, the nuclear hormone receptors, sets the basal metabolic rate, how many calories your body needs to run itself at rest. It's also the case that the development of fat cells and the storage of fat into those cells is under hormonal control as well, and they're regulated through a family of nuclear hormone receptors that have the terrible name peroxisome proliferator activated receptor. Basically, these are receptors for fats that do different things. And the most key one is the receptor called PPAR gamma, which is considered to be the master regulator of fat cell development. If you express PPAR gamma in a stem cell, that stem cell is now a pre-fat cell, a pre-adipocyte. If you activate PPAR gamma in a pre-adipocyte, it now becomes a fat cell, an adipocyte. And if you activate PPAR gamma in an adipocyte, it stores more lipid. So this is a very, very important gene in the whole process, and we'll keep returning to this again and again throughout the talk. I'd next like to introduce the concept of endocrine disrupting chemicals. So in the body, the endocrine system produces hormones in certain locations throughout the body, largely in the hypothalamus, in the pituitary, and these hormones travel through the bloodstream and have effects on distant organs. And an endocrine disruptor is a mixture of chemicals, or exogenous chemical or mixture of chemicals that interferes with how hormones work. This is the most recent definition that's been adopted by the Endocrine Society, which is the largest professional society that deals with the endocrine system. And these endocrine disruptors can be the wrong signal. They could also cause the loss of the signal, so the absence of the hormone's action, or to have the signal at the wrong place at the wrong time. And it's important to note that hormones in your body work at very, very low concentrations. The estrogen receptor and the thyroid hormone receptor work at parts per billion, very tiny amounts of hormone. So it stands to reason that the chemicals that disrupt those can also do so at very low levels. So how are we exposed to endocrine disruptors? Well, these are persistent organic pollutants in the environment. They're come in through the diet as pesticides, fungicides, herbicides. We're exposed to them at great amounts through personal care products, through cleaning materials, and through food packaging, among others. And the Endocrine Society came out with a statement in 2009, actually the very first scientific statement of this nature in their almost 90-year history, that laid out the evidence for the action of endocrine disrupting chemicals and whether or not they were harmful. And the conclusion was that the evidence for adverse reproductive outcomes from endocrine disruptor exposure is very strong. It's beyond question. The data is so voluminous. And there's mounting evidence for effects on the thyroid system, the neuroendocrine system, the way that your brain chemistry functions, and obesity, metabolism, insulin, and glucose homeostasis. This statement by the Endocrine Society was adopted by the American Medical Association shortly thereafter. And very significantly, the European Union Parliament just a few months ago voted to direct the European Commission, which is the regulatory agency, to present bans on the restrictions or the use of endocrine disruptors by June of 2015 by almost a 5-to-1 margin. And that's very different from what's happening in this country. So what kinds of chemicals are endocrine disrupting chemicals? This is a partial list which comes from the National Institute of Environmental Health Sciences. And you can see on the left side of the list there's a number of herbicides and fungicides and insecticides that can all be endocrine disruptors. Perhaps the most famous of these is DDT, which has been banned some years ago. And to me, it's not so surprising that these classes of chemicals might have adverse consequences on living things because they're designed to be toxic to life. Herbicides, insecticides, fungicides are designed to kill living things. So it's not terribly surprising that they have adverse consequences on systems they weren't designed to target. But to me, it is quite surprising that industrial chemicals, that the plastics that we use, that detergents, that the coatings, that these chemicals should have endocrine disrupting effects. That's really completely unexpected. And if I summarize, you can see in this little blurb on top of the slide that these fall into many, many categories. And we encounter these quite a bit. Perhaps very worrisome is the presence of endocrine disrupting chemicals in personal care products, which we willingly and at great amounts put all over our bodies. And you can see in this slide that many of the personal care products that we use all the time have endocrine disruptors in them. And you can see the different ones in different personal care products. And to me, it would be very useful if some company were to make endocrine disruptor-free personal care products. If you doubt, or if you take the industry position that these chemicals are not getting into us, you should read this book by Rick Smith and Bruce Laurie with the silly title Slow Death by Rubber Duck. But what these guys did is they exposed themselves to everyday chemicals and then measured the concentrations in their bodies. So they plugged in an air freshener and they sat in the room with it for a couple days and measured the concentration of chemicals in their body. They used lotions and creams and measured the chemicals. And the conclusion was that these chemicals get into your body. Not everyone agrees. For example, the position of industry is that there are very little risks from endocrine disruptors. And I like this slide on the right. It says, we in the business community prefer a cautious wait-and-see approach over needless media scaremongering, while he's grown very large breasts. So the position is that there are no endocrine disruptors, and if there are, that they're not very dangerous, and that we really shouldn't worry about it. And I would present the counterexample. This is a table from a paper by Tracy Woodruff and her colleagues, showing at the top that reproductive diseases and disorders of a great variety have increased over the last 50 years worldwide. And at the bottom of the table, you can see that reproductive function around the world has also decreased over the last 50 years. So it could be the case that it's just a coincidence that we've put lots of endocrine active substances in the environment and we have disturbances in endocrine regulated diseases, or there may be something to it, and that's a conclusion that you need to make for yourself. I think the evidence is quite clear. So how does this come into obesity? So what I and my colleagues have been working on for the past 10 years or so is whether endocrine disruptor mediated disturbances in the endocrine system have anything to do with the development of fat cells and the accumulation of fat in the body. And that was a silly cartoon. This is reality. Unfortunately, American kids are getting fatter and fatter all the time. And the sad reality is that children that are the size of these often become adults that are this size. And it's really not a good thing to have a population of adults that can't get around and require lots of extra health care. It would be nice if we could prevent this from happening so that everyone was healthy and happy. So here are the players in our story, the nuclear hormone receptors. These are a family of what we call ligand-activated transcription factors, which means that they turn genes on or off depending on whether the hormone is there. At the top on the left, you can see that there are many receptors which we knew from biochemical experiments. In there you see the glucocorticoid receptor, the estrogen receptor, the testosterone receptor. And according to the Environmental Protection Agency, endocrine disruptors are chemicals that interfere with the testosterone receptor, the estrogen receptor, or the thyroid hormone receptor. I'm going to tell you a story about chemicals that disrupt two other receptors, the peroxisome proliferator activated receptors and the retinoid X receptor. But in principle, there's no reason why endocrine disruptors can't encompass or act on any or all of these receptors. And the revised definition of endocrine disruptor from the Endocrine Society, which I told you earlier, really considers that possibility. So Lynn mentioned that we came up with something which we called the obesogen hypothesis, and according to me, obesogens are chemicals that inappropriately stimulate the development of fat cells or storage of fat into those cells, that disturb the balance of fat tissue in your body, or alter the control of appetite and satiety that ultimately leads to weight gain and obesity. And there's evidence for chemicals that act in all these different ways. So the earliest evidence that there were such things as endocrine disruptors that affected obesity came from the work of Retha Newbold at NIEHS. And you can see the two mice on the lower right hand of the slide. And the mouse on the left was exposed to just normal water, and the mouse on the right was exposed to 5 parts per billion of diethylstilbestrol for the first 5 days after birth. And that mouse, despite having only that brief exposure to chemicals, got really quite obese. And it's also important to note that if you give those animals 5 parts per million, 1,000 times more of diethylstilbestrol, they actually lose weight. They don't become fat, they become smaller than the mouse on the left. And this is an example of what we call a non-monotonic dose response, and industry and regulatory agencies just discount that possibility out of hand. So Rita has shown that the soy estrogen genistein and other people have shown that bisphenol A also gives the same effect. We know that there are drugs that make people fat. So thiazolidine-dione anti-diabetic drugs, Actos and Avandia, which target our friend PPR gamma, make people fat. We know that the level of phthalates in the urine of men in the studies from the Centers for Disease Control are correlated with waist diameter and insulin resistance. And in the past 5 years or so, many studies have come out that have linked the presence of one chemical or another with obesity in human epidemiological studies, even after you allow, if you adjust for the amount of fat that people have. And we know that there are chemicals that cause fat cells to differentiate in cell culture models. And these include phthalates and bisphenol A, and over to the right, our favorite molecule, organotins. So I think that the existence of such things as obesogens is really quite plausible. It's not something fantastic or unrealistic, but it's something that's really quite believable, and there's a lot of evidence. So this slide shows our favorite obesogen, tributyltin, which is in the lower right. And tributyltin is famous as an endocrine disruptor because it causes what's called imposex in mollusks and snails. And you can see the snail on the left, there's a little red circle, and in this little red circle is, inside that circle is the penis, and on the snail on the right, you can see that it's grown to absolutely gigantic proportions, and as you can imagine, the parts don't fit anymore, and these animals are sterile. I became interested in tributyltin when it was shown that it could sex reverse fishes from female to male. And we asked the question, which hormone receptors would be targets for those organotin effects? And what we found was that tributyltin activated at very, very low levels, at the levels at which you find it in the environment, RXR and PPR gamma, which are important for the development of fat cells. The TBT induced cells in culture to become fat cells, and if we fed it to mice, the mice became fat. That's published a long time ago, so I won't show you any data, except this one little bit here that shows the fat pad around the testis from a 10-week-old mouse, and you can see on the left is the control mouse, and it's around about 300 milligrams, and on the right you can see that it's about 15% bigger in the mice that were exposed to TBT. They didn't eat any different diet. They didn't do anything except be exposed to tributyltin. So we've been asking in recent years, how does this happen? How does this exposure to tributyltin cause weight gain? You could imagine, and the standard argument would be, well, the mice just eat more. Well, in fact, they don't eat more. They actually eat less. Well, perhaps we've altered the ability of the fat cells in these mice such that they tend to store fat. Or perhaps we've made more fat cells or more pre-fat cells. And the reason we think that is because, hello? There are classes of cells called mesenchymal stem cells, which are precursors to many other cells in the body, including bone, cartilage, and fat. These cells are known to become fat cells after you give them rosiglitazone, Avandia, PPR gamma. So the hypothesis was that TT is induced genesis, making mesenchymal stem cells into fat cells. And this slide shows our mesenchymal stem cell, and you can see it become bone cells or it can become fat cells. And the key player here is our friend PPR gamma. So we gave these mice either vehicle control, or we gave them rosiglitazone, or we gave them tributyltin, and we asked the question, what's the fate of these cells when we grow them in culture? And I'll just summarize. Once we give the chemicals to the animals and isolate the cells, that cells which came from animals who were treated with rosiglitazone or with TBT made many more fat cells. They expressed markers of fat cells. They expressed these genes which mark fat cells. They didn't make bone. They didn't express markers of bone differentiation while at the same time making fat cells instead. I'm going to skip that. So what we've been wanting to know recently is, how does this happen? How do we get from a pre-fat cell to a fat cell? And we know that our friend PPR gamma is involved, and that when you activate PPR gamma, you turn on all these genes that make these cells into these cells. And we know that when we give the maternal exposure to tributyl-10, many of these genes are expressed at higher levels, and we believe that means there's an epigenetic change that makes these genes be expressed more. Here's an example of one such epigenetic change. This is the regulatory region of a gene called fatty acid binding protein. And if you take away methyl groups in the promoter of this gene, you will have it be higher expressed. So what you can see here is that, indeed, when we expose them to tributyl-10, there are less methyl groups in this promoter, and there's more expression. So that leads me to the last and perhaps most important part of the talk, and that's when we've asked, are the effects of this exposure to tributyl-10 permanent? So in order to ask that question, we gave mice tributyl-10 in their drinking water while they were pregnant, and we gave them either solvent control, Avandia, or 3 different doses of tributyl-10. And I want to make the point here that these are very, very low doses. These are the kinds of doses you might see in the environment, and these are below the level that's considered to have no effect. So we exposed pregnant moms, and we took the babies, and we sacrificed some, and we mated some, and we got F2 and went out to F3. And what we did is we looked at body weight, the size of the fat tissue, the size of the fat cells, the gene expression in the stem cells, and we also looked at the liver. If you see an effect in F1 and F2, so in the babies or their offspring, this is called a multi-generational effect. And that's because these animals were exposed. So mom was exposed, the fetus was exposed, and inside that fetus are germ cells, which will become the F2 generation. So they're all exposed. In F3, these animals were never exposed to chemical. So if you see an effect in F3, it's called a transgenerational effect, and it's considered to be permanent. That means it will last forevermore. So what did we find? We found, when we looked at the size of the blobs of fat in the body, that some of the fat depots in F1 were a little bit bigger. This is around the kidney. This is between the shoulder blades. But if we looked in F2, you can see that the fat depots are all much bigger, and you see the same thing in F3. So this effect persists into the F3 generation. If we look at the size of the fat cells, you can see that in all generations, the fat cells are much bigger. So not only have we made bigger blobs of fat, but it consists of bigger fat cells as well. So what about gene expression? Well, this is the expression of genes that are important for the development of fat cells. Here's our friend PPR gamma. You can see that it's up. Fatty acid binding protein 4. Lipoprotein lipase is the most important gene in fat storage. And you see not only is it expressed higher in F3, but it's basically off the charts. Same with FABP4. And zinc finger protein 423 regulates PPR gamma. So all of these genes that are important in the development of fat cells are expressed at much higher levels in cells from animals that were exposed to tributyl-10. We also saw something interesting when we looked at the livers of these animals. And you can see the livers were noticeably more pale. And when you slice the livers and when you stain them with a dye that reveals fat, and that's this dye here where you can see red called oil red O, you can see that in the animals that were exposed to tributyl-10 that in all generations there is a lot more fat in the liver. Now remember, these animals were eating a normal diet. They were not exposed to a high-fat diet. These were just exposed to tributyl-10 when the F0 mom was pregnant, yet we see through all the generations fatty livers. Interestingly, if you look in females, you see the same thing, except that rosiglitazone causes fatty livers in F1 females, but not in males. If you look at gene expression in these animals, you see that in addition to having more fat in the livers, they express genes that make fat. PPR gamma, SRBP1C, fatty acid synthase, they're all expressed at very high levels in all 3 generations. These genes that regulate the burning of fat are also increased, PPR alpha, acyl-CoA oxidase, and this gene here that coats the blobs of fat, called FSP27, is also very highly upregulated. So not only in the stem cells and in the fat, but also in the liver, we've caused a permanent transgenerational change that predisposes these animals to accumulate more fat. So let me summarize what I've told you. The first is that organotins are very potent activators of these 2 important hormone receptors at the kinds of levels that we're likely to see. The tributyltin makes cells become fat cells in cell culture model, and when we expose animals to tributyltin during development, in our hands we can make more fat cells in 2 species, in mice and in frogs. Other people have shown in fishes and in rats that you get the same effect. So it's very likely that you will get the same effect in humans. The effects of maternal exposure are transgenerational. That is, the effects on the size of the blobs of fat, on the size of the fat cell, on gene expression in the stem cells, are all transgenerational. There's multiple ways that tributyltin can accomplish this. I've shown you the data for RXR and PPR gamma, but there are other possibilities as well that could add to the effect. So people always ask me, are organotins making me fat? Well, the answer is that if you expose adult animals of all kinds to tributyltin, they get fat. And we know in humans that drugs that activate PPR gamma make people fatter. So why would you expect that a chemical that targets the same receptor would have a different effect? You would expect the same effect. We know that the prenatal exposure to tributyltin makes mesenchymal stem cells become fat cells. So the most important question is whether humans are exposed to sufficient levels of tributyltin for us to worry about. And the answer is we don't really know because no one is measuring. What we do know is that polyvinyl chloride plastics are 3% by weight of organotins, 0.1 molar, really a lot. Now luckily, most of that is monobutyltin and dibutyltin. And the plastics industry is very careful to say we never, ever, never put tributyltin deliberately into plastics. And that's correct, they don't. Except that dibutyltin is about 95% pure. And the other 5% are, guess what? Tributyltin, because dibutyltin is made by knocking the side groups off of tetrabutyltin. So it's what's called a non-intentionally added substance. They don't mean to put it there, but it's there anyway. Tributyltin is still found in seafood. Triphenyltin is still used on crops in the United States. And there are some studies that show detectable levels of organotins in some populations, but this is something that needs to be studied in much greater detail. I think it's safe to conclude that human exposure to organotins can possibly reach levels that are sufficient to activate these receptors. So is the environment making us fat? Well, I'm not sure I would say that, but what I would say is that the evidence indicates that there are such things as obesogens which can affect the way animals and humans respond to calories and predispose them to become fat. So how many such chemicals are there? I've shown you data for tributyltin, and triphenyltin works the same way. There's pretty good data in animals for phthalates and perfluoroalkyl compounds. There's a huge data set on estrogens. We know a lot about nicotine, actually from human studies, and other components, air pollution and benzopyrene, which comes from smoke and burned foods, are also obesogenic. You perhaps have heard my colleague Rob Lustig shouting from the rooftops about the obesogenicity of fructose. It's a very big concern. There are other chemicals. We've shown there, other people have shown organophosphate pesticides. We've shown that some fungicides are all obesogens. So how many are there? Is that it? Are there just these 10 or so? Are there 20, 100, 1,000, 1,000,000? probably the number is going to be some hundreds up to a few thousands, but I think we have a long way to go before we know that. We know very little about how much any one of us have been exposed to. We know some of the molecular targets of action. We know about RXR and PPR gamma. There are other chemicals that make animals fat that we don't know what the pathway is. We don't know about all the critical windows of exposure. When is it that chemicals can make us fat? We don't know a lot about how prenatal exposure alters the adult phenotype. We don't know a lot about the epigenetics or epigenomics of this. So the implications for human health, I think, are clear. The first is that diet and exercise, so too much food and too little exercise, are insufficient in and of themselves to explain this obesity epidemic. And the biggest data supporting that are that Rob Lustig has an epidemic of obese 6-month-old babies. And you can argue all you want that teenagers and adults eat less or eat more and exercise less, but that argument fails when you talk about 6-month-old babies because they eat until they're full and they don't exercise very much. Obesogens exist and they inappropriately stimulate the development of fat cells and storage of fat into those cells. And the best evidence for that is that there are pharmaceutical obesogens. There are drugs that make people fat. Diazolidine make people fat. Atypical antipsychotics, tricyclic antidepressants make people fat. And as you would expect, there are chemicals that target the same pathways that have the same effects. Organotins do it, estrogens. There's a whole host of chemicals which we know make people fat. Prenatal exposure to obesogens reprograms exposed animals to be fat despite normal diet and exercise, as best as we can determine. And we believe that this results from epigenetic changes that alter the fate of stem cells such that they make more fat cells. Here's a silly cartoon I found, how his epigenome interacted with butter pecan to make him obese. I think seriously that the identification of obesogens says even more strongly that we need to shift the paradigm from treating obese people to preventing children from becoming obese in the first place. Because the reality is that once a person becomes obese, no matter how hard they try to lose weight, 90% of them will gain the weight back. So we need to prevent children from becoming obese, and the problem will go away. I'd like to also raise the issue of chemicals with transgenerational effects. So we've shown that tributyltin has transgenerational effects. That means maternal exposure is permanent. We know that from Mike Skinner's work that vinclozolin, which is a fungicide, and a variety of plastics and hydrocarbons from jet fuel and DDT all cause transgenerational permanent effects. And there's recent data that show that bisphenol A that comes from plastics and thermal paper and recycled paper and food packaging alter social interactions in animals and modify gene expression. So the existence of chemicals that have transgenerational effects really changes the debate, I think. We know that endocrine disruptors act at low environmentally relevant doses to adversely affect many organ systems. They're widespread in the diet. We're all exposed. We know that prenatal or early life exposure can have permanent effects on the health and susceptibility to disease. And we know, very importantly, that effects of prenatal exposure can be transmitted to subsequent generation, that is, your children, your grandchildren, your great-grandchildren, and beyond. So I think this really changes the debate, that we cannot simply say that if we take these chemicals out of the environment, assuming that the effects will go away. The effects can persist, so it means that we need to take more care rather than less care in this process. Lastly, I'd acknowledge the people who did this work. Most of the work I told you about was done by 2 postdoctoral fellows in the lab, Severin Kirchner and Raquel Chamorro-Garcia, and we gratefully acknowledge funding from the University of California, the US EPA, and the National Institute of Environmental Health Sciences. And that is all I have to say, and I'd be delighted to answer whatever questions people might have.
Thank you so much. I found that extremely interesting, and I'm sure everyone else did as well. I guess that I'll start off, Bruce, with a couple of questions. And so how do you think that the science that you've raised here, what kind of impact do you think that can or should have on policy in this country?
So that's always a tough question.
Usually, engage themselves with policy, but I'm just interested in your opinion here.
So my opinion is that it's had, as best as I can detect, it's had very little impact on policy in this country.
Mm-hmm.
And that's unfortunate. It's had a very huge impact on policy in Europe.
Uh-huh.
So as I mentioned, the European Parliament voted 5-1 to direct the European Commission to get rid of endocrine disruptors in the environment and in the food. So they have to do that now, and that's caused a firestorm of controversy. Industry is trying like crazy to fight that because they don't want to stop selling their endocrine disruptors and make different chemicals instead. They want to keep on doing things just like they've been doing them.
Mm-hmm. Mm-hmm.
So we would hope that we learn from the Europeans and adopt some similar policy decisions, but that remains to be seen.
Okay. So I have some good questions here. Have there been any epidemiological studies on pre-postnatal exposures to DES, smoking, or genistin from formula in humans, and whether there were impacts on obesity rates in later generations?
So smoking, there have been many, many studies. They all focus on the F1 generation. There are almost no studies that look at subsequent generations because it takes too long and there just aren't those cohorts of people. So DES, you would imagine, is the cohort could exist. DDT, you would imagine, could be studied. So there are some long-running cohorts, like the one in Berkeley that Barbara Cohn is now in charge of, that they're probably in the F3 generation. So these studies can be done given funding and interest. They're not inexpensive, and the money has to come from somewhere to do the work.
Okay. So that's a hint, as a shout out to somebody. If there are agencies on the line I know, and some companies if they want to. So shout out for some funding that's necessary to study things. So has anyone experimented with possible interventions such as increased exercise to see whether obese mice can lose weight following a prenatal exposure to obesogen? The question is really about whether obesogen-induced obesity is modifiable in the animal model.
That's a very important point. So I think that it's critical to understand that obesogen-induced obesity is really just a propensity, right? It's a change in the way your body handles calories. So it means if you've been exposed and had your body reprogrammed by your mom's obesogen exposure, it means your battle is harder than it would otherwise be in the current environment, right? So given the current calorie-rich environment, it means you have to try harder. You have to exercise more. You have to really be careful about what you eat. But if you go off and live under famine conditions, then you're going to do much better. And we hope that we never get to that situation again. Mm-hmm. So have studies specifically been done on obesogen exposure? No, they haven't. And that's something that requires interest and funding, and someone will do those studies, I'm sure.
But you're not absolutely doomed if you have these exposures. You can control it based on personal decisions as well. I
don't believe that you're doomed. I think that the data are thin, but mm-hmm. I believe, based on how we understand the mechanisms, that you are not doomed. But what you are doomed is to have to fight harder. Mm-hmm. And that raises the issue of free will, right? So is it fair to say, here's an obese person and here's a normal weight person? Can you say to that obese person, you really, even because of the way your body is, you have to work much harder to not be obese, and I demand that you do it? is that a fair thing to say? I mean, that's a discussion we need to have.
Well, I think it's a really critical public health issue because there have been a lot of campaigns and programs directed at obese individuals and children in particular, which really do focus on shaming and, blaming the victim in a lot of ways. And I think it's really important to have in terms of understanding how to integrate lifestyle components as well as what the scientific, other aspects to it are.
Right. This is a fundamental flaw in the tradition and in what I call the orthodox wisdom. The calories in, calories out, all calories are the same, all calories can be part of a balanced diet, eat less calories and you'll be fine. If it were so simple, no one would be obese because no one chooses to be obese. Nobody wants to be obese, except for the one lady whose name I forget, who's trying to be the fattest woman in the world. Aside from her, no one wants to be obese, yet many of us are. So it's despite our free will and our interest that people are becoming obese. And that needs to be recognized, and we need to do everything we can to make it easier for people to not become obese. And getting rid of this silly calories in, calories out argument is the first step in that.
So I have another question. Is there evidence of synergistic effects of the many obesogens we are exposed to every day?
There is currently no evidence for that, but it's because the studies haven't been done. There aren't so many of us studying obesogens at the moment, and there are a few grants I'm aware of that have been given to study mixtures, and in a few years we will know the answer to that, and I would say it will probably go in multiple directions. There will be some chemicals that are synergistic or additive, and there will be some chemicals that cancel out each other's effects. And it just depends on the particular chemicals that you're talking about.
I think we have time for a few more questions. I have one here that says, what positive outcomes are possible if exposed prenatally?
Positive outcomes from prenatal exposure to obesogens.
I guess that's the question.
That would be a tough one. I don't imagine that there should be positive outcomes under normal circumstances. But if you take the example of the thrifty genes and you take some animals that were exposed to obesogens and some animals that weren't, and you calorie restrict them, you put them under semi-starvation conditions, the prediction would be that the animals that were obesogen exposed will survive better. Now that's just my hypothesis. Someone needs to do that experiment.
The next question is, how should evidence of endocrine disruption be treated in a risk-based paradigm in order to address a policy goal that seeks a ban on EDCs? In Canada, such evidence is considered as one line of evidence. Is this an appropriate way to consider endocrine disrupting potential?
As the one line of evidence as harm?
Yeah, I think so. I mean, there's.
So this raises an important issue that I've talked about in other venues, and that's the law in this country. So in the civil law, we decide cases by what's called preponderance of the evidence, so that the evidence is more in favor of one side than in the other side, and then we decide in favor of the side for which there's more evidence. In the criminal law, the decisions are made based on what's called beyond a reasonable doubt. So beyond the doubt that a reasonable person would have when looking at these set of facts. In the chemical law, we need to prove that a chemical causes harm to humans beyond the remotest shadow of a doubt. And that is ridiculous. And that is the law that the regulatory agencies operate under at this moment. That needs to change.
That's the risk-based paradigm, right?
Yeah.
Yeah. And well, the questioner was talking about Canada in particular, but I think it's very much the same. So basically, you're saying that a risk-based paradigm is not going to be the most appropriate in trying to deal with endocrine disrupting chemicals.
Well, the problem is how to calculate the risk. And EPA has formulas that they use for risk, and the risk formula nowhere incorporates the magnitude of the risk, the likelihood that harm will happen.
Words in your mouth about that. I didn't phrase that correctly.
No, that's fine.
Sorry.
And we can talk for hours and hours about the chemical law and how it is and how it should be changed, and there are many things wrong with it, but the very biggest thing wrong with the law is that the people doing the risk assessment assume that all chemicals must have what's called a monotonic dose response. That means linear. That means that the amount that as you decrease the amount of chemical, the effect decreases, or as you increase the amount of chemical, the effect increases linearly. And there is not a single thing linear in nature except gross toxicity. Everything else is nonlinear. Every system, every natural living system is nonlinear. So why we would assume linearity in risk assessment is beyond me, and it's beyond most endocrinologists. Endocrinology is not linear.